Welcome to the FSL course. My name's Mark Jenkinson and I'll be talking to you today about a range of different topics in the background of neuroimaging so that you can understand how they relate to the analysis and get the most out of the FSL course. What we'll be covering is a range of different things from some basic anatomy through to some acquisition and then a little bit of the analysis. Most of the analysis we're going to cover in the main FSL course, but you need to understand how these different aspects interact with analysis so that you can actually get the best out of your acquisitions, know how to do the right analysis, and how to interpret your results at the end of the day. What we'll be covering in this introduction is listed here, and we'll spend most of the time looking at the three main modalities that we'll cover during the main FSL course. They are structural, diffusion, and functional MRI. So let's start by talking about basic anatomy. Now this is really for people who haven't done a course before in biology or neuroanatomy and have come from a different background. So they might have come from a maths or physics or engineering background like myself, and you just need to know some fundamental things in order to understand some of the things that we're going to talk about later on. So it's going to be quite quick and quite basic, but it is really essential for those of you who haven't looked at this before. The first thing to say is that the main cell that we're going to be interested in is the neuron. And you can see an instance of a neuron here. But actually, when we're looking at the gross level of the anatomy, we're looking at things like you can see in the postmortem dissection in the bottom left, or in the MR image of the top right. And here you can see just a few different shades of tissue. So you can see that there are some brighter parts and there are some darker parts. And these are named in gross anatomy as grey matter and white matter. They relate to the different parts of the neurons in this way. In white matter, what you have are the axons, and so most of the axons are concentrated in the white matter. They are the things which connect the different neurons together. They're the outputs from one neuron and how it relates its signal to the others. In the grey matter, which is darker in the dissected slice that you can see on the bottom and happens to be darker in the MRI slice that you can see at the top, though that can change, and we'll see some instances later where that's different in different MRI acquisitions, you'll get predominantly the neuronal cell bodies. And that actually makes the tissue darker because it doesn't have so many axons. It's actually the property of the axons which makes the tissue lighter. But the main thing is that these are the two main types of tissues that we see. They both have neurons in, just different parts of the neurons. And they're a rough description. It's predominantly neuronal cell bodies in the grey matter, but not exclusively. There are also axons there. It's mainly axons in the white matter, but there are actually other cells as well, which are present everywhere. So these are rough descriptions which are very helpful in neuroimaging, but also very helpful in gross neuroanatomy, and so they're common terms. Another thing that we'll also refer to is some of the fluid-filled spaces, such as you can see here, which outlines the lateral ventricles. These are filled with a fluid called cerebral spinal fluid, or CSF. And this we will sometimes loosely refer to as a third tissue. So you'll often hear us referring to different tissue types being grey matter, white matter, or CSF. And that's just a loose way of us having these three different convenient types of things that we can see in our MRI images. The actual interconnections of the neurons is very complicated. They're densely connected. There are lots of connections between the dendrites, the inputs to the neuronal cell body, and the axon terminals, the outputs from a neuron onto lots of dendrites from lots of other neurons. And you can see just here a little example of what that might look like. It's actually the axon which is of a lot of interest because that's the predominant thing that we see in the white matter, and it's actually attacked in a lot of neurodegenerative diseases. One of the things about the axon is it's surrounded by a thing called myelin, or the myelin sheath. And that comprises lots of fatty molecules and is actually one of the reasons that we see a different look to the tissue when we're looking at our postmortem dissections, and a different MR properties of the tissue. And it's actually one of the things which gives the MR properties, which is good because it allows us also to look at changes in myelination and what happens when these cells are attacked or degenerate for some reason during the disease process. And so it's a very interesting and very important factor in MR. And that's all I'm going to say about basic anatomy. As I promised, it is basic. It's really there just so that you understand what these sort of terms mean and the fact that then they are catch-all terms. They are not precise. They are rough terms describing predominantly neuronal cell bodies in the grey matter, for instance. But that changes in other areas. So the next topic I'll talk about is just a range of different applications that we can have in MR neuroimaging. And I'll cover this because it'll be familiar to some people, but actually you may not be familiar with the whole range. For some people, it might be quite new. So if you're new to the area, then you might not have seen some of these things. We don't expect you to get all the details out of these things. It's really just to get an idea of the scope of what you can do with neuroimaging. And so what we cover, and the main concentration of what we will look at in the course, is structural imaging, diffusion MRI, and functional MRI, which has two different types. So what I'll do now is describe one example study in each of those so that you get a feeling for what is actually looked at in each of these cases. So the first one is structural MRI. This is an example of a study which was done in 2000 looking at taxi drivers. And the fact that taxi drivers, particularly in London, can't just rely on their sat-navs. They actually have to learn about the layout of the streets and the one-way system. They have to actually really build up a mental map so that they can navigate efficiently around the city. And what this study did was scan people at different stages of how long they'd been a taxi driver and look to see whether there were any changes that they could detect in the structural MRI, which was reflected by how long that they'd been actually driving. And what the structural MRI is good at showing you is it's good at showing you structure boundaries between different parts of the brain, or just quantifying the amount of, say, grey matter which is present. And that's what this study did. It looked at the amount of grey matter and whether there was a change which was in relation to the length of time that people had been driving taxis. And you see the results here. It actually found one part of the brain called the hippocampus where there was a relationship, as you can see at the bottom there, between how long people had been driving taxis and the amount of grey matter. And that was an early example of structural plasticity in a healthy adult brain, showing you that the healthy adult brain isn't just static, it doesn't just stay the same, it actually responds. You can actually have an increase of neurons, an increase of grey matter in response to tasks, in this case learning and navigating around the city. Another example is diffusion MRI. Diffusion MRI looks at the microstructural properties in the white matter predominantly. And you can tell a lot of different things about the white matter from that. You can look at directionality of the connections, the anatomical connections of these axons within the white matter, how they connect one part of the brain to another, or you can look at very local microscopic properties of how the myelin has changed or how the axon density may have changed. This is an example of a study in Asperger's syndrome where it looked at comparing a couple of different groups, one with Asperger's and one without, to look at whether there was a noticeable change in the properties of the white matter. And what you see here is an area where all of the parts in red to yellow show a statistically significant difference between the two different groups in the major white matter tracts. The parts in green show the white matter tracts where there weren't any changes that were picked up between the groups, at least at the statistically significant threshold that was used here. So this is a way that we can look at whether there are changes between different groups and whether there are other things going on in the white matter, so separate from the grey matter that we looked at with the structural MRI. Functional MRI comes in two different flavours. One of them is task, and that's when you're actually looking at the dynamics of the brain, you're putting the participant in the scanner and getting them to do some particular task. In this case, we're looking at a stroke study, and it was looking at people who'd suffered a stroke and how they responded to therapy, and particularly what areas of their brain were responding when they were having to do manipulations with their hands. One from the unaffected side and one on the affected side from their stroke. And what this showed was that there was actually changes in the areas and the amount of activity which was present after the therapy as compared to before the therapy. And so that's a way of monitoring the efficacy of the therapy, but also understanding what it's doing. Is it recruiting different areas from, say, the ipsilateral side of the brain, as opposed to what would normally be used, which is the contralateral side? So that's one way that we can look at function in the brain, giving a task and looking at response to task. A separate way is to look at a resting state fMRI, or functional MRI. And in resting state, we're actually not giving the participants any particular task. We're looking at spontaneous activity which occurs in the brain and looking to see whether there are different parts of the brain which are connected based on the fact that they have similar spontaneous activity. And if we do that, then we can look at networks in the brain and we can do all sorts of different analyses. In this particular example, we were looking at how things had changed in a group situation between young, healthy adults with or without a particular genetic risk factor. So all the individuals were healthy, they just happened to have a particular gene, ApoE4, and when they had the gene, then there was actually an area which was coming up as differently connected as to the group that didn't have the gene. And in that situation, we can see here, it might be a little bit difficult to see on that slide, but you actually see some things in the hippocampal region. Again, something which is known to be affected in Alzheimer's, but these individuals are decades before they would actually see any changes, any clinical symptoms due to Alzheimer's. So this shows a very early change in the way that their brain is functionally working, and the functional connectivity, the information that we can get out of looking at resting state fMRI. But it's not just about using individual modalities in isolation. There's a lot to be gained from combining different modalities together. And now I'm just going to have a couple of examples to illustrate that. But actually, it's an important thing which is becoming more and more prevalent in the field. So this is a surgical planning example where we're combining information that we get from diffusion imaging and that we get from task-based functional imaging. What you can see here is an example of a patient that has a tumour. That's the big thing in blue. What we use the functional task for is to get information about where the motor response is happening. So what part of the brain is actually responsible for motor activity in that patient. And we can map exactly where that is. Then we use diffusion to trace out where is the connection between that part of the cortex, that part of the grey matter, and how that connects down through the brainstem into the spinal cord. Because you need both of those in order to actually be able to move fine. And that's crucial information for the surgeon so that they can then go in and try and remove the tumour, but spare the part of the cortex which is actually responsible for the movement and also not sever the connection of that part of the cortex with the rest of the body. So having both of those bits of information in this instance is crucial. But it's not just in clinical context where that's useful. A lot of research can gain from having multiple modalities used. Here's an example of a structural MRI and diffusion MRI, where we're looking at the structural MRI to give us information about what areas have changed between different groups, in this case a Parkinson's disease group and a matched group of controls. And we can see that there are certain areas which are predominantly affected in Parkinson's, and we can use diffusion information to trace out where those axons go, what parts of the brain are they connected to, so that we can get more information about other subsystems which might be affected as a consequence of this change that we see in the subcortical regions, or maybe vice versa. Maybe it's changes remotely which are responsible for some of the changes that we see in the subcortical regions. And that's the kind of information that we can leverage by having a couple of different modalities used together. And that's all I wanted to talk about in terms of the sort of general broad overview of what you can get out of different modalities of MRI. So that's useful to get a broad picture of things, and if you're new to the field then you might not know all of that. If you have been doing a particular type of investigation in the field, then also some of that might not be familiar with you. Again, this is something that you don't need to know all the details about, it's just to give you a flavour for the kinds of things that you can do. If you are new to the field, then we would recommend that you would watch this talk very carefully and maybe re-watch it later before you start the course so that you can get the most out of the FSL course. The main thing is to get a grasp of the fundamentals that we're trying to get across. So what I'm going to do now is talk about and spend most of the time in the talk going through some of the acquisition modalities and understanding some of the principles upon which that's based, which actually feed into the analysis. So we'll be talking about both things in conjunction. Before I do that, just a couple of words about MRI analysis in general. So one of the things to realise is that MRI, regardless of what kind of it is noisy. There is always noise in MR to the point that it's a problem. If we did nothing about it, we would have difficulties knowing what was a real finding and what was a finding which could just be driven by noise. So we're always having to do something about it. We can get great information out of MRI and we have good ways of dealing with the noise, but it's always there, it's always something to be thought about. The other thing is that MRI scanners are very variable, they're very configurable. You've already seen the kinds of things that we can get out. We can get out information about diffusion, about structure, about function, all from the one scanner. And that's great. But it's also configurable within those types. There are different types of diffusion scans you can get, different types of structural, different types of functional scan. And that means that our analysis has to be able to cope with that. It has to be able to cope with the breadth and the different changes of kinds of acquisitions that we can get out. So there's not a single analysis that we can do, there are lots of options available. It also has to be based on statistics, because that's what deals with the noise. And there's also, even if we know exactly what kind of acquisition that we've got, there's often more than one right way to do an analysis. There's often several different types of analysis with different pros and cons associated with them. But that's not to say that there aren't wrong ways of doing analysis. There are definitely wrong ways, and you need to avoid those. And what this course is going to try and teach you is to explain the aspects so that you can make good choices about how to do analysis, about how to acquire images well, and how to interpret the results correctly. It's going to give you enough information that's relevant for understanding how to run the analysis without giving all the details of the inner workings. But you do need to know enough to avoid doing analysis the wrong way. Okay, so within each of the main modalities that I'll talk about, there are various different points, mainly concentrating on how the measurement is done and also how that is going to interact with the analysis and the kinds of artifacts that we're going to see. So let's start by talking about structural MRI. So here there are three different types of images that you can see: T1 weighted, T2 weighted, and proton density. They look very different in terms of how bright different things are. So you can see that the fluid filled space in the CSF is very dark, and the T1 weighted is very bright, and the T2 weighted. They all, however, tell you about gross brain anatomy. They're not telling us about the direction of the wiring in the white matter. They're not telling us about functional activity within the brain. They're just telling us about the gross structure in the anatomy. There's still a variety of different types of images that we can get. So you can see three here. There are some more we'll talk about later. But even within those, there are different resolutions that we can get, and there's a different amount of noise that we can get depending on the parameters that we use. And one of those things is how long we're going to acquire the images for. So you can get very quick images, 3 minutes or so, but that will limit you to your resolution and how noisy the images are. Or you can spend a lot longer taking images, up to say 15 minutes or even more for a single image, to get very high quality imaging. So there are lots of different types of sequences, but the T1 weighted is the most common. So we'll talk mainly about that. The kinds of things that you can do with a T1 weighted image are summarised here. So one of the things that we can do is that we can look at the different tissue types, the grey matter and the white matter CSF, as I described before. There's a thing called segmentation. We can also look at how to find different structures, things like the hippocampus or the outer cortex. We can model the cortex and all of its folds. We can measure things like thickness, and we can also look at changes in its thickness, or we can look at changes in the local amount of grey matter, like we saw in the taxi driver study before. What structural MRI is based on is measurement of predominantly water molecules, and that's common across not just structural but all of the MRI that we're talking about. MRI is capable of getting information back from different nuclei. The ones that we're interested in are the hydronuclei, and the hydronuclei in water molecules are the ones which are most present in the brain. There are also hydronuclides in other molecules, such as fatty molecules, and you do get signals back from those as well. But inside the brain, it's really about the water. And the water is the thing that we will be imaging and that we'll be looking at most. And there's a lot of information that we can get from that water, because it's not just about the amount of water. So the density of water is very important, and we call that proton density, because it's the hydronuclei within that count. But actually also the magnetic properties that water has, because it's a magnetic technique, and there are certain properties that the water molecules have depending on what kind of environment they're in. And that's crucial. The environment that they're in changes two main properties called T1 and T2. You don't need to know what they mean at the moment. There will be a MFR physics lecture during the course which will unpack that a bit more. These two main properties are the things you will hear it talked about. You've already heard me mention T1 weighted imaging before. And in this case, these are the three things: the density of the water and the T1 and T2 relaxation properties, which are determined by the environment. There are lots of things in the environment which do change these relaxation properties. And that can be things like how fast the water molecules can tumble, but predominantly things like what other molecules are there, how are they restricted, what kind of other electrical and magnetic interactions do they have. All of these things determine the kind of signals that we get back. And that all goes into the mix in terms of getting our intensities, which is what we use to infer tissue type. And it's actually a really complicated mix of things. So knowing the intensity doesn't tell us much about what an individual aspects of that microscopic environment are like. But all of those put together determine our intensity and give us different intensities in the grey matter and the white matter, which is crucial for us to be able to use these structural images and get lots of information from them. Just knowing that and what I've said so far gives us a little bit of an idea about some of the limitations that we have in MRI. So one of them is that we don't directly measure grey matter, white matter and CSF. We don't get an intensity value of 2 for grey matter and 1 for white matter, for instance. It's not like that. We get an intensity value of 900 or something like that. And then that doesn't actually tell us much on its own. It's whether the next tissue along has a bigger intensity or a lower intensity, which would tell us whether that particular tissue is more likely to be white matter or grey matter or CSF. So that is coupled with saying that it's not quantitative. The values of the intensities themselves in isolation don't tell us anything. It's the relative values of intensities between a particular voxel and a voxel next to it. A voxel is just a three-dimensional pixel, if you haven't heard that term before. And it's the relative intensities between these and the different tissue types which really matters. So it's not quantitative, but we can get a lot of information out of these images. And the T1 and T2 relaxation properties determine this intensity, and they are very different between grey matter and white matter. However, they also vary within grey matter and within white matter. The variations are much smaller, they're much more subtle, and they represent variations typically in the biological substrate, in what the proportion of neuronal cell bodies versus axons would be, for instance, would determine small changes in T1 and T2 relaxation properties. And that can be interesting in and of itself. In MRI, we also can't distinguish bone from air. So we look at water, as I've said, and we really don't have very much water in bone, and so we don't get much signal back from it. And so we get very dark areas, which could be bone or they could be air. Both of those give very little signal back. We can't easily distinguish between the two with MRI. We also have variable contrast. So by contrast, I simply mean the difference in intensities between two different tissues, and that can be easy to see in some areas and not so easy in others. And it can be, for instance, very good at the outer cortex, but quite poor in the subcortical areas, and that's not uncommon. And so you'll see that not all grey-white interfaces are the same, and that in order to get the most out of your image will depend on what you want. If you're particularly interested in subcortex, you might need to actually adjust to get better contrast there in your acquisitions. We also find that a single sequence doesn't tell us all. So if we're interested in just a healthy cohort, it's very common just to get a T1 weighted sequence. But actually, if we've got a cohort with some clinical pathologies, we will need to get something else. And we often get other images such as T2 weighted images, proton densities, or other kinds, because they highlight different aspects of pathology much better than the T1 weighted images do. And then we also have the limitations of artifacts and noise. So here I'll talk a little bit about some of the artifacts that we see. So one of the things that you'll always see is noise. I've talked about that already. And there are two aspects of that, contrast to noise and signal to noise. And we talk about SNR being a signal to noise ratio or CNR being a contrast to noise ratio. In the two images that you can see here, the one on the left-hand side is much noisier. It has a much worse signal to noise ratio or contrast to noise ratio. The one next to it has much better signal to noise ratio or contrast to noise ratio. That means it has less noise. And you can just see in there that it's easier to see the boundaries. It's easier to find where the grey matter is and where it stops and becomes white matter to get out the information that we want. That's partly because this image took a lot longer to acquire than the one on the left. But the crucial thing is to understand that there are lots of options and you will get lots of different images out of MR if you look at different studies, and they will vary a lot in terms of how much signal to noise ratio they have. The difference between signal to noise ratio and contrast to noise ratio is simply that contrast is about the difference in intensities between neighbouring tissues. And so you want that to be large versus the noise in order to be able to see the difference between tissues. Signal to noise ratio doesn't care about that, it's just the average level of the signal versus noise. The other thing that we have is changes in spatial resolution, and that's associated with partial voluming. And so you can see on the right-hand side an example where we've got a very low spatial resolution, which means that we've got a big voxel size. So in the images on the left-hand side, we have a 1 millimeter voxel, which is very typical for structural imaging. on the one on the right hand side, we've got a 3mm voxel. And what happens there is that we have a greater mixture of tissues within that voxel typically. So there's some grey matter and some white matter and maybe some CSF as well. And what happens then is that the intensity you get back from that is a mixture of those. And that means that we don't see a clear boundary. We see sort of intermediate intensities when we go from one tissue to the other, but also it means that the actual just fundamental resolution, the size of the voxel is so big that we can't clearly delineate precisely where the boundaries are. So we suffer when we change resolution in terms of trying to find things like structural boundaries. But that's often a trade-off that we have to make. And there's a trade-off between the resolution that we have, the amount of noise that we have, and the amount of acquisition time that we have to use in order to get these images. And you need to make the right kind of trade-off in order to do the kind of things that you're most interested in. There are other artifacts that we have. This is an example of what's called an RF inhomogeneity, or also called a B1 inhomogeneity. And this results from the fields which are radio frequency fields, so the RF, which come from the head coil. Ideally, we would want that to be completely uniform throughout the head, but it never is. The head coils can't be designed to give a completely uniform field. And so what happens is that in certain areas you end up with the intensity being a lot lower than it should be. That is, the image looks darker than it would be. Or it could be the other way around. The image looks brighter than it should be. And so you can see examples of both of those in the images here. Darker on the left-hand case, brighter at the back on the right. And that happens more when you've got high field scanners like 7 Tesla scanners, or when you've got head coils with lots of different coils in them. So you might have heard of 16 channel or 32 channel coils. Number of channels is the same as number of coils, and the more that you have, normally the less smooth the bias field is, and the more that you might see these intensity variations across the image. Now this is something which is always present in the images, but something which we can very well deal with in the analysis stages later on. So we don't normally worry too much about that acquisition unless it's particularly bad. But there are other sorts of artifacts which are problematic and we do need to sort out and try and eliminate. One of them is motion. Motion is always a problem in MRI. You'll always get some motion of the subject in the scanner. The best thing to do is to explain to them that they should not move, to try and make them relaxed and to sort of pad their head so that they're comfortable and can't move too much. But nonetheless, even small amounts of motion can be a big problem for MRI. And here you can see some examples of what it looks like in a structural image. You have lots of these sort of lines, these ringing lines next to the edge of the brain. And that's quite a lot of motion in order to induce that in structural imaging. You need a lot less motion to be a problem in functional and diffusion imaging, as we'll see later. But any motion is a problem and should be tried to be minimised at source. So you really need to try and deal with that whilst the scanning is happening. It's something that we can't as easily clean up later on in the analysis. There are also other things which happen as a consequence of imperfections in the hardware. Ghosting is one of the things which is common. It's not normally as strong as this, it's normally very, very mild. When it's mild, we just put up with it. It's something that doesn't cause too much of a problem. Wraparound, however, is something where it's going to be equally as bright as the rest of the brain. And here you can see an example where the back of the brain has ended up at the top of the image, where it shouldn't be. And that's simply because the field of view was made too small for that head. And that's really a mistake that was made when that scan was being taken. And that's something which can be fixed at the point where that scan was being acquired. And so there's really no reason that we should ever have wraparound. And wraparound in an instance like this is a real problem and would just make that scan unusable, because that overlap between the back of the head and the front of the brain means that we don't know what the intensity of the brain should be at that point. And I'm afraid there are even more artifacts which can occur. So there are things like RF interference, where you get some signals coming through the door or the patch panel. So that's normally a case of something not actually being sealed correctly. Or you can have RF spiking, which could just be a static discharge from a blanket, or it could be some electrical sparking, which is happening in some of the equipment. They both cause very obvious things in the images, as you can see here. And there are actually other artifacts which can occur too. The good news is that they're really uncommon. And actually they're very obvious when you look at these images. And so they're easy to identify. What we recommend for actually identifying these is a very easy to use, low cost artifact detection device, which is simply your eye. Just look at the data. That's all you need to do in order to see whether you've got some of these nasty artifacts. If you do, then just seek somebody out who's experienced, who's got some expertise, and they can tell you what's the best course of action to take at that point. Because it will vary depending on the kind of the artefact that you get. But it's really quite uncommon. There are some artefacts like the bias field, which we always have, and the noise, which are always going to be there. But these other artefacts, they're very uncommon. If you do see them, seek out some advice. The good news is that most of the time that doesn't happen. You look at your images, there aren't any obvious artefacts, so you can move on to the analysis. And there are various different things that we can do in the analysis for structural imaging. So one of them is brain extraction, and that's just removing the non-brain tissue from the image, because actually we're just interested in analysing the brain. We can do a segmentation of the tissue type, so that's grey matter, white matter, or CSF, as I was talking about before. We can do a segmentation of structures, so that might be hippocampus or thalamus, or it might be the outer cortex and all of the folding associated with that. And there's also registration or alignment of different individuals, and we do that in every study, whether it's between different images of the same individual to account for the amount of head motion that might have occurred during a scan, or what happens all the time is in research studies we have groups of individuals and then we have to register each of those or align them together so that we have a common anatomical coordinate system. We can actually say that this point is the same point in everybody's brain so that we can get some consistency in our findings. All of these different kinds of analyses give us some information about the amount of grey matter or the structural boundaries, other things like that, the cortical thickness. What they all have to feed into is statistics. As I said, we always have noise, so we always need to do statistics. And we're going to set everything up in the general linear model. We'll cover that in the main course. But the thing to understand is that we are always going to have to do something, whether it's functional or diffusion or structural. There's always statistics involved. And it's often to look at groups of individuals, compare different groups, or to look at one group and how it compares to something like performance or learning in a task, like in our taxi driver study. And we can use it in structural imaging to look at things like changes in grey matter, changes in the shape of a structure, or changes in the cortical thickness. All of these use statistics in order to analyse what's going on across a group of individuals, taking into account both the noise in the individual MR scans, but also the biological variation across that population. I'll talk now a little bit about some tips for acquisition of structural MRI. So for a T1 weighted image, that normally has the best trade-off between SNR, the amount of noise that you would have, the amount of time that it takes you to get the acquisition, and the resolution that you get. We normally aim for about 1 millimetre resolution in our voxel sizes, and T1 weighted images normally represent the best images to go for. However, always follow the advice of the people who actually run the scanner that you're scanning on, because there's a whole range of different kinds of sequences which are out there, and they can be tweaked in different ways by changing different parameters to be better or worse at certain things. So explain to them what you want to do, and they'll be able to tell you what they know about what's been optimised, what works well on that particular scanner. It's much better than looking at literature, much better than talking to somebody who scans somewhere else. Really talk to the people who work at the place where you'll be scanning, because they know what will work well there. If you've got a population where you know that you can't stop movement, so it might be a very elderly population, might be a sick population, might be a very, very young population, there's a limit to how good the scans you can get are. But what you want to avoid is the situation where you're doing a 10-minute scan and that participant has moved badly in the middle of it and you have to throw the whole thing away. One of the options that you've got is, instead of doing one long 10-minute scan, is to do a bunch of shorter scans, so 5 2-minute scans for instance, and then average together all the ones which are good. You can throw some of them away if there's been particularly bad motion in them, but still keep some and get a good enough result at the end of the day. So that could be a useful tip if you are working with difficult populations. If you're interested in subcortical areas, then again pay attention to tweaking the contrast in those areas, because that might not necessarily have been done with the sequences that you're using, because it might be being used by somebody else who is mainly interested in cortical contrast, the outer grey matter. There's also an option to turn a thing called fat suppression on, and we would recommend that if you can, because that helps us with brain extraction. It actually removes the fatty signals, which can result from things like the membranes outside, the bone marrow, things which are very close to the brain and could look a bit like brain tissue, which just makes brain extraction more difficult. If you can get rid of those at the acquisition stage by using fat suppression, it doesn't have to be total fat suppression, just a little bit of fat suppression can help and make things a lot easier. Importantly, go for isotropic voxels. By what I mean by that is a voxel size which is the same in every direction. So a 1 millimetre by 1 millimetre by 1 millimetre voxel is an isotropic voxel. It doesn't matter if it's a little bit off, it can be 1 by 1 by 1.1, that's fine. But try and avoid the case where you've got really small in-plane resolution and very thick slices. So you might have something like a 0.5 by 0.5 in-plane voxel size and then a 4 millimetre thick slice. That's bad for the kinds of quantitative analysis that we're going to do because it biases the ability to find different borders in different directions. It's not uncommon clinically, so that's why I'm warning you now, is that is a sequence which you may well come across. Try and avoid it if you can and go for something close to isotropic. There's also another option on the scanners to do a thing called upsampling, and that's a bit like on your digital camera. You have an option often to go for something like an effective resolution of 20 megapixels, where there's actually the real number of megapixels on the camera might only be 10. That is a way of creating an image which has a resolution which is better than was actually captured by sort of doing some tricks in some mathematics. It can look nice visually, but it causes all sorts of relationships within those intensities which are problematic for automatic analysis, quantitative analysis that we're going to do. So try and avoid that. Don't do the upsampling on the scanner. Okay, so that's talking about some of the kinds of things that we can get out of structural MRI. There are other kinds of structural MRI acquisitions that we can get, as I mentioned before. Here are another three. So one is flare, one is white matter null, and one is double inversion recovery. Each of these is an example of what happens when we suppress one particular tissue's intensity. So in the flare, we suppress the fluid. It's fluid attenuated, so that's actually suppressing the intensity from the CSF. In the white matter null, the key is in the name, it nulls the white matter, it gets rid of the signal from that. In the double inversion recovery, it gets rid of both white matter and CSF signal. And these can be really helpful for highlighting different pathologies which exist, or for looking at very fine detailed anatomical structures. And these sequences are quite common if you are doing anatomical work or if you are working with certain types of pathologies. There are also other kinds of sequences. Here are another three: susceptibility weighted or quantitative susceptibility imaging. That highlights aspects of iron content within different tissues. There's also magnetization transfer, which is another way of highlighting aspects of pathological tissues typically, or veno and angiograms, which actually allows you to look at the vessel network, the blood supply within the brain. And so these are some of the kinds of sequences that we can get, the kinds of acquisitions which sit within the realm of structural MRI. There are lots of different things that we can get, and they relate to different aspects of the tissue that we're sensitive to. So as I said, the susceptibility-related ones are sensitive to iron. If your pathology is something where iron accumulation is important, then that's a great kind of image to get, because it will give you extra information on top of what you could possibly see in something like a T1 weighted image. And so each of these has a different aspect of the tissue that it's able to be sensitive to. So one of the great things about MRI is that how flexible things are, how many different options we've got of highlighting different aspects of the tissue. And we can also measure things which are not just about the biological tissue. We can measure things about the scanner itself and the fields that it creates. So you'll hear things like the B0 map. B0 is the name for a particular magnetic field which exists within the scanner. It's actually the big field, the 3 Tesla field or the 1.5 Tesla field that you have through your scanner. And you might also hear that called as a field map. You'll hear that later on in the course. A B1 map or an RF map, again, is mapping a different kind of field, the one that was causing our bias fields, our changes in the inhomogeneities that we had before. And we can get sequences which measure those in addition.